Now, let's get a closer look.
The small intestine is the longest portion of the gastrointestinal or GI tract. It's approximately 6 meters long in adults, and it's the main site for both digestion and absorption of food in the body. Although the majority of the small intestine shares similar histological structures, it's still divided into 3 main segments: the duodenum, jejunum, and ileum. The most proximal segment of the small intestine is the duodenum, and it's also the shortest segment as well. The duodenum connects with the jejunum at the duodenojejunal junction. The jejunum is approximately 2.5 meters long and will gradually transition to the ileum, which is the last segment of the small intestine. The ileum is usually about 3.5 meters long and leads to the cecum of the large intestine at the ileocecal junction. Similar to the rest of the GI tract, the wall of the small intestine has 4 main layers: the mucosa, submucosa, muscularis propria, and in the small intestine, the outermost layer is a layer of connective tissue called the serosa. In these images, the serosa is only present in the image of the duodenum. Although each segment of the small intestine shares similar overall structures, there are still quite a few differences between each segment that can be seen with light microscopy. Even at low magnification, we can see that the finger-like projections, or villi, that extend into the lumen of both the duodenum and jejunum are very tall and slender compared to the villi of the ileum, which are significantly shorter, broader, and whose tips are flatter compared to the villi of the duodenum and the jejunum. Let's take a closer look at the mucosa of the duodenum. The villi of the entire small intestine are lined with enterocytes, which are simple columnar cells with microvilli. The microvilli increase the surface area available to the enterocytes for absorption. The pale cells within the villi are the goblet cells, which secrete mucus for lubrication and physical protection of the intestinal epithelium. The lamina propria is the loose connective tissue that supports the enterocytes and forms the middle or core of each villus. The crypts of Lieberkühn, or intestinal crypts, are glands found at the bases of the villi. The crypts contain stem cells that slowly differentiate into the cells that form the epithelial lining of the small intestine, which include both the enterocytes and goblet cells. The differentiated cells move up the villi as new cells are continually formed underneath them, and within a week, the cells will reach the tip of the villi where they're shed into the lumen, similar to the separated epithelial cells seen at the top right of this image. Some of the crypts in the small intestine have cells with very eosinophilic, or pink, granules. These cells are called Paneth cells, and they help provide an innate immune defense against intestinal pathogens. And finally, underneath the villi, crypts, and lamina propria is a thin supporting layer of smooth muscle called the muscularis mucosa. If we look at a section of the jejunum, we can see that the mucosa shares the same characteristics we saw in the duodenum's mucosa, including the crypts of Lieberkühn, Paneth cells, enterocytes with their microvilli, goblet cells, lamina propria, muscularis mucosa, and the shedding of the epithelial lining. Now, if we look at a section taken from the ileum, it has the same characteristics as the duodenum and jejunum, but the mucosa of the ileum can be differentiated by its shorter, broader villi, as well as Peyer's patches. Peyer's patches are masses of lymphoid tissue within the lamina propria that are only seen in the ileum. Also unique to the ileum are lacteals, which are the white spaces within the cores of some of the villi. These spaces actually represent lymphatic capillaries that absorb dietary fats from the intestinal lumen. The next layer of the small intestine underneath the mucosa is the submucosa. Portions of the mucosa can still be seen at the top of these images in green. The submucosa consists mostly of dense, irregular connective tissue all throughout the small intestine, which is dark pink when the tissue is stained with hematoxylin and eosin. The duodenum submucosa can be distinguished from other segments of the small intestine by unique glands called Brunner's glands, which are only seen in the duodenum and esophagus. They can be identified in images as a thick layer of coiled tubular glands lying just beneath the muscularis mucosa. In the duodenum, they secrete alkaline mucus into the lumen of the small intestine in order to reduce the acidity of the gastric juices and partially digested food coming from the stomach. Near the top of this image, we can see that the glands will occasionally pass through the muscularis mucosa in order to release their secretions into the crypts between the villi. If we go back and look at the images of the submucosa from the jejunum and ileum, we can see cross-sections of nerves that are part of a network of nerves called the submucosal plexus or Meissner's plexus. These nerves are responsible for innervating the mucosal goblet cells and the smooth muscle of the muscularis mucosa. We can also see a few large blood vessels within the submucosa that still have red blood cells within their lumens. All segments of the small intestine have a muscular layer that surrounds the submucosa called the muscularis externa or muscularis propria. This layer actually consists of two main layers of smooth muscle arranged in different directions. The inner layer has a circular or circumferential arrangement around the wall of the small intestine. The muscle cells of the outer layer have a rounder appearance compared to the inner layer because they have a longitudinal arrangement that runs parallel to the length of the small intestine. So in these images, we're only seeing cross-sections of these muscle cells. Between the two layers of smooth muscle is another network of nerves, but these nerves are called the myenteric plexus or Auerbach's plexus. The myenteric plexus innervates the smooth muscle cells of the muscularis externa that surround the nerves. Looking at the same image, we can see a portion of the serosa, which is the fourth and outermost main layer of the small intestine. The serosa is a thin layer of loose connective tissue that allows the small intestine to move more freely within the abdomen and contains blood vessels, nerves, and lymphatic vessels as well.
Now, let's get a closer look. The large intestine consists of the appendix, cecum, colon, rectum, and anal canal. The colon is mainly responsible for reabsorbing water and electrolytes from the feces within its lumen. The colon is continuous with the rectum, where the feces is stored before defecation. Similar to the rest of the large intestine, the colon has four main layers: the mucosa, submucosa, muscularis propria, and a surrounding serosal layer of connective tissue that isn't seen in this image. Even at low magnification, we can see that the colon's mucosa at the top of this image doesn't have the distinct long villi or finger-like projections that would normally be seen in the small intestine. Taking a closer look at the colon's mucosa, the lumen of the colon is seen at the top of the image, and the first layer of cells lining the mucosa is the epithelium of the mucosa. The epithelium consists of two types of cells, enterocytes and goblet cells. The enterocytes, or absorptive cells, are the simple columnar cells with microvilli. They're also called the absorptive cells because of their main function of absorbing water from the colon lumen. And the goblet cells are responsible for secreting mucus. Although the cells aren't clearly seen in this image, the mucus they produce is easily seen as the globular structures that are stained dark purple from the hematoxylin and eosin stain. The surface epithelium is continuous with straight, unbranched, tubular glands called the crypts of Lieberkühn. Unlike the crypts in the small intestine, these crypts extend through the majority of the mucosa, from their openings at the intestinal surface all the way to the muscularis mucosa along the deepest portion of the overall mucosa. It may not always appear to be continuous on histological slides, because the path of the crypts may not always travel along the same plane as the section of tissue taken from the colon. The superficial portions of the crypts will typically have a higher concentration of enterocytes, and the deeper portions will have a high concentration of goblet cells. The tissue found between the crypts and the epithelium is the lamina propria, which consists of many types of immune cells, including plasma cells, lymphocytes, eosinophils, and macrophages. And finally, the deepest layer of the mucosa is a thin layer of smooth muscle called the muscularis mucosa. Beneath the mucosa is the next major layer of the colon, the submucosa. This layer consists mostly of dense, irregular connective tissue, but also contains blood vessels, lymphatic vessels, and the submucosal or Meissner's plexus. Meissner's plexus is a network of nerves that innervate the goblet cells in the mucosa as well as the smooth muscle of the muscularis mucosa. In this image, there's a relatively large cross-section of a bundle of nerve cells that are part of Meissner's plexus. We can also see a number of large blood vessels and a lymphatic vessel in the upper left of this image. Surrounding the submucosa is the next main layer of the colon wall, called the muscularis propria, or muscularis externa. The thick muscularis propria allows the colon to have strong peristaltic or wave-like contractions to help move the feces through its lumen. The inner layer of smooth muscle is arranged circularly, or circumferentially around the colon's wall, and the outer layer of smooth muscle is arranged longitudinally along the length of the colon, but it's also arranged into 3 long bands of muscle called tinea coli. Between the 2 layers of muscle, we can see cross-sections of nerves from another network of nerves called the myenteric or Auerbach's plexus, which is responsible for innervating the muscle fibers of the muscularis propria. Also, some portions of the colon will have an additional thin layer of connective tissue that surrounds the muscularis propria called the serosa. Alright, as a quick recap. The colon has 4 main layers: the mucosa, submucosa, muscularis externa, and the outermost layer of serosa. The mucosa is comprised of an epithelium, lamina propria, crypts of Lieberkühn, and muscularis mucosa. The submucosa consists of dense irregular connective tissue, blood vessels, lymphatic vessels, and Meissner's plexus. The muscularis externa is a thick layer of smooth muscles with an inner circumferential layer and an outer longitudinal layer that's organized into 3 bands of smooth muscle called tinea coli. Between the layers of smooth muscle is the myenteric plexus, which is the network of nerves responsible for innervating the smooth muscle of the muscularis propria. And the final, outermost layer is the serosa, which is a thin layer of connective tissue that also contains blood vessels and nerves. Now, let's get a closer look. The stomach is an expanded portion of the gastrointestinal tract, or GI tract, that partially digests food by breaking it down mechanically and chemically in order to form a pulpy acidic fluid called chyme. The stomach is divided into 4 anatomical regions: the cardia, fundus, body, and pylorus. But the stomach is only divided into 3 histological regions: the cardia, fundus, and pylorus. That's because the fundus and the body are histologically identical, so both regions are called the fundus when referring to their histology. The cardia is a small area surrounding the opening to the esophagus which contains cardiac glands that secrete mucus. The fundus is the largest region histologically, since it also includes the body of the stomach as well. This region of the stomach has fundic or gastric glands that secrete digestive enzymes such as pepsin and a protective layer of mucus. The pylorus is the most distal region of the stomach before reaching the pyloric sphincter. This region will have a combination of pyloric glands that secrete mucus and neuroendocrine cells that secrete gastrin. Similar to the rest of the GI tract, the wall of the stomach has 4 main layers: the inner mucosa, submucosa, muscularis propria, and outer serosa. Although some portions of the GI tract have an outer layer of adventitia instead of serosa. This low-power longitudinal section of the stomach was taken from the cardia of the stomach. Now, if we take a closer look at the mucosa of the cardia, we can see that the surface of the mucosa has a simple columnar epithelium with many invaginations that form millions of gastric pits. The gastric pits will comprise about a quarter of the mucosa's thickness. At the base of these pits, they join with multiple tubular cardiac glands that secrete mucus that protects the esophagus from gastric reflux, and is also a part of the stomach's gastric juice. The cardiac glands extend all the way to its underlying layer called the muscularis mucosa, which is a thin layer of smooth muscle that is still a part of the mucosal layer. The next main layer of the cardia is the submucosa, which consists mostly of dense, irregular connective tissue, but also contains larger blood vessels like the ones we can see in this image. On the right of this image, we can see a portion of the overlying mucosa, and on the left is a portion of the next main layer called the muscularis propria. The entire stomach's muscularis propria is unique when compared to the rest of the GI tract, because it has 3 layers of smooth muscle instead of only 2 layers. In this image, we can see portions of the inner oblique layer on the right side of the muscularis propria, closest to the submucosa. The middle layer is the circular layer, and the outer layer is a longitudinal layer of smooth muscle. Because this tissue sample was sectioned longitudinally, the outer layer of smooth muscle is the layer that shows the long shape of the muscle fibers the best, and the middle layer will have muscle cells that appear the most circular. The outermost layer of the cardiac stomach is the serosa, which consists of loose connective tissue with large blood vessels, adipose tissue, lymphatic vessels, and nerves. In this image, the large blood vessels are easy to identify because of the red blood cells that can be seen within their lumens. Alright, let's move on to the fundic stomach. This low-power image allows us to see three of the main layers: the mucosa at the top, the submucosa, and the muscularis propria at the bottom. The outer serosa isn't present in this image, though. The prominent fold in this image is called a rugae, which is formed by both the mucosa and submucosa. The rugae allow the stomach to expand after food or liquid is consumed. The submucosa, muscularis propria, and serosa are very similar to the cardiac stomach. The fundic mucosa may look similar to the cardiac mucosa, but the glands present in the mucosa are different. Instead of cardiac glands, the gastric pits join with fundic or gastric glands, which consists of three major cell types: parietal cells, mucous neck cells, and chief cells. The gastric glands are also broken down into three regions: the isthmus, neck, and base of the glands. The isthmus consists mostly of parietal cells, which can be identified in this high-power image by their very eosinophilic or pink cytoplasm, and their large round nuclei that are centrally located. Parietal cells can also be found in the neck and base of the glands, but overall, there are more present in the isthmus and neck. These cells are responsible for secreting gastric acid as well as intrinsic factor, which is a glycoprotein required for the uptake of vitamin B12 in the small intestine. The neck of the gastric glands consists mostly of parietal cells and mucous neck cells. The mucous neck cells can be found in between the parietal cells as smaller, pale, basophilic cells that are also seen in the base region of the gastric glands. In addition to parietal cells and mucous neck cells, the neck also has stem cells, but they can be difficult to identify using only an H&E stain. Although, when mucosal cells are damaged, the stem cells will multiply in order to replace the damaged cells, making the stem cells easier to find histologically. For example, gastritis and peptic ulcers are common conditions that can cause significant mucosal damage, resulting in an increased number of stem cells within the mucosa. If we move down to the base of the gastric glands, parietal cells and mucous neck cells will still be present, but the base also has very prominent peptic or chief cells, which are responsible for secreting digestive enzymes such as pepsinogen. These cells have a granular cytoplasm and are very basophilic, resulting in their dark purple appearance. Enteroendocrine cells such as G cells are also present at the base. These cells are responsible for secreting hormones, including gastrin, but they aren't easily identified with an H&E stain. Alright, let's move on to the last and most distal region of the stomach, the pylorus. Although the serosa can't be seen in this image, the serosa, muscularis propria, and submucosa are all very similar to the fundus and cardia, but the mucosa of the pylorus does have quite a few unique characteristics. The mucosa of the pylorus has more prominent gastric pits that comprise about half the thickness of the mucosa instead of only a quarter or less. As a result, the pyloric glands are generally shorter, have more branches, and more coils when compared to the glands of the fundus or cardia. The majority of the pyloric glands are mucus-secreting cells that are similar to the mucous neck cells. The glands also contain cells that are not as easy to identify, such as a smaller number of parietal cells, stem cells, and G cells, which secrete gastrin. Now, let's get a closer look. The skin covers and helps protect the body from its environment. It's also known as the integument, which is part of the integumentary system. The integumentary system also consists of structures that are derivatives of the skin, such as nails, hair, and glands within the skin, like sweat glands. The skin can be split into two general types, thick skin and thin skin. Thick skin has a much thicker outer layer of epidermis that helps to protect the body in places where it's regularly exposed to slightly harsher physical conditions. That's why it's typically found in places such as the palms of the hands and soles of the feet. Thick skin can also be differentiated from thin skin because it doesn't have hair follicles or oil-producing glands called sebaceous glands. It does still contain sweat glands, though. The overall thickness is often more than 5 millimeters, whereas thin skin is typically only between 1 to 2 millimeters thick. Thin skin has all the same structures as thick skin, except it has a thinner epidermis and additional structures such as hair follicles and sebaceous glands. If we compare histologic images of thick skin and thin skin at low magnification, we can see the three main layers that make up the skin. At the top of the image is the surface epidermal layer, or epidermis, and that's the part that's significantly thicker in thick skin. Hair follicles are only found in thin skin, and as we can see in this image, they're located in the middle layer called the dermis or dermal layer. The third and deepest layer is called the hypodermis, which consists mainly of adipose and loose connective tissue, although this image of thick skin doesn't include the hypodermis. Now, if we look just at the epidermis, there are some sublayers. Starting from the base of the skin is the stratum basalis, spinosum, granulosum, lucidum, and corneum at the surface. The stratum lucidum can only be seen histologically in very thick skin. If we take a closer look at the epidermis of both thick skin and thin skin, we can see that the stratum basalis is only a single layer of cuboidal or columnar germ cells. These cells are attached to a basement membrane that's normally too thin to see under the microscope. These cells have a high level of mitotic activity, dividing to produce all the cells that migrate up to the surface of the skin. In both thick and thin skin, the cells within the stratum spinosum synthesize a fibrous protein called keratin, which is why the cells themselves are called keratinocytes, even though they also produce other proteins as well. This image of thick skin was taken while using a 60x oil immersion objective. Oil immersion objectives allow for higher resolution images to be seen through the microscope. All the skin cells of the epidermis are keratinocytes, except for the germ cells, which are found in the stratum basalis. The keratinocytes are attached to each other by strong cell-to-cell structures called desmosomes, which are more easily seen in this layer compared to the layers above the stratum spinosum. The desmosomes are found in between the cells and look like tiny needles or spines. As the keratinocytes mature, they're pushed up closer to the surface by newly formed cells below, and gradually become flatter in appearance as well. Above the stratum spinosum is a layer of 3 to 5 flattened cells that contain dense basophilic keratinohyalin granules within their cytoplasm, which is why this layer is called the stratum granulosum. In thick skin, the lighter or more lucid layer of cells seen above the stratum granulosum is the stratum lucidum, which is typically only 2 to 3 cells thick. The keratinocytes in this layer will have lost their nuclei and organelles, although they're still attached to one another by desmosomes. This layer won't be seen above the granulosum in tissue sections taken from thin skin. Now, the stratum corneum is the outermost layer of dead keratinocytes from the underlying stratum lucidum. So these cells also don't have nuclei or organelles, but in this layer, the cells are almost completely filled with keratin filaments. This layer consists of approximately 15 to 30 layers of the squamous keratinocytes. Over time, the desmosomes in this layer gradually break down, which causes the cells to shed or desquamate from the surface of the skin. In thick skin, the stratum corneum is often thicker than all of the other epidermal layers combined, whereas in thin skin, this layer is much thinner when compared to the rest of the epidermis. All right, let's move on to the dermis, or dermal layer that sits below the epidermis. The junction between the two layers is irregular, forming many projections of the dermal layer called dermal papillae. The corresponding invaginations of the epidermal layer are called epidermal ridges, and the combination of the papillae and ridges is referred to as the papillary layer of the dermis. The dermal papillae help improve adhesion between the epidermal and dermal layers. This image is from thick skin. If we compare it to thin skin at the same magnification, the dermal papillae in the thin skin are noticeably smaller, which makes the papillary layer thinner as well. Sensory receptors called Meissner's corpuscles are found in the dermis. These elliptical structures are found inside dermal papillae and are positioned perpendicular to the surface of the skin. They function as mechanoreceptors, which mean they sense or respond to physical pressure or movement of the skin. But they're also specialized to detect light touch and low-frequency vibrations better than the mechanoreceptors found in deeper layers of the dermis. When textured objects are moved across the skin, they create the low-frequency vibrations that are detected by Meissner's corpuscles. Alright, let's zoom back out to the low-magnification images of thick and thin skin. The papillary layer is the outer portion of the dermis that includes the dermal papillae, but the rest of the dermal layer is called the reticular layer. This layer is much thicker and contains a lot of dense irregular connective tissue, but this is also where many of the specialized structures of the skin are also located, such as sweat glands and sensory receptors called Pacinian corpuscles. In addition to these structures, thin skin will also have hair follicles, sebaceous glands, and arrector pili within the reticular layer. The eccrine sweat glands are the coiled tubular glands and ducts that produce and secrete sweat onto the surface of the skin. When seen histologically, we can only see cross-sections of the tubular structures. As a result, the gland looks like a collection of circular structures that each have a central lumen. The walls of the sweat glands consist of a simple or stratified cuboidal epithelium. These sweat glands are found in both thick and thin skin. In this image of thin skin, we can see the secretory component of the sweat gland in the bottom of the image, as well as part of the ducts as they travel to the surface of the skin. Because the ducts are not producing fluid, the ductal cells have a cytoplasm that's more dense. This means that the ducts will typically stain a darker purple when compared to the secretory component of the sweat glands. The Pacinian corpuscles are the mechanoreceptors in the dermis that are better at sensing coarse touch, pressure, and high-frequency vibrations when compared to the Meissner's corpuscles. Sensing high-frequency vibrations allows the skin to distinguish the difference between different fine surface textures, such as the difference between paper and glass. The Pacinian corpuscles are large round structures that have 20 to 60 concentric layers or lamellae of modified Schwann cells. That's why these corpuscles are also often called lamellar corpuscles. The lamellae surround a central inner pulp that contains unmyelinated axons. In this image, we can see that the corpuscles are also surrounded by an outer capsule of connective tissue. Alright, let's move on to hair follicles, which are only found in thin skin. The base of each strand of hair and hair follicle has a hair bulb, which contains specialized basal epithelial cells that produce the keratinocytes that eventually become the hair shaft, as well as the inner root sheath. Hair shafts are made up of dead keratinocytes that have lost the majority of their cellular detail. The inner root sheath immediately surrounds the hair shafts within the hair follicle and is split further into two sublayers. The outer layer is called Henle's layer, which is a layer of simple cuboidal cells, and the inner layer is called Huxley's layer, which consists of a single or double layer of flat cells. Surrounding the inner root sheath is the external root sheath, which contains layers of cells that are continuous with the epidermis. Separating the external root sheath from the dermis is a hyaline layer without cells, or an acellular layer called the glassy membrane. The dermal papilla is the connective tissue and network of capillaries that helps bring nutrients to the growing basal cells and projects into the base of the bulb. In this image of a hair follicle, we can also see the sebaceous glands nearby that produce and secrete an oily and highly lipid fluid called sebum. The cells of the sebaceous glands are called sebocytes. They have a central nucleus, and their cytoplasm is filled with small fat droplets that give the cell a foamy-appearing cytoplasm. The glandular cells then drain into converging ducts that typically empty into the upper portion of the hair follicle. Just below the base of the sebaceous gland, we can see part of a small muscle. These muscles are able to move the hair follicles they're attached to, causing the hairs to stand on their end when a person is cold or scared. Now, let's get a closer look. The prostate gland is a large and dense exocrine gland that's responsible for secreting a white alkaline fluid that makes up about 30 to 50% of the seminal fluid volume. It's the largest accessory sex gland in the male reproductive system, measuring approximately 3 centimeters in length and width, with a height of 5 centimeters. The average weight of a normal prostate gland is about 11 grams. Due to its similar size and shape, it's sometimes compared to a walnut. The gland surrounds the most proximal portion of the urethra, or prostatic urethra, just below the bladder, and consists of 30 to 50 branched tubuloalveolar glands, such as the one seen in this low-power image of the prostate. The glands all drain into converging ducts that eventually empty into the prostatic urethra. The alkaline fluid that's secreted by the prostate gland also includes various small molecules, fibrinolysin, citric acid, and the clinically important prostatic acid phosphatase, or PAP, and prostate-specific antigen, or PSA. Normally, only a small amount of PSA will leak into the prostate's vasculature and circulate in an individual's blood. But a high level of circulating or serum PSA is a sign of abnormal prostatic tissue, which could be caused by prostate cancer, inflammation, or benign prostatic hyperplasia. Because of this association, a PSA serum level is often used as a tumor marker for prostate cancer. And even after a patient has had their prostate cancer removed, PSA is used to monitor for a possible recurrence of the prostate cancer. PSA and PAP immunostains can also be used on tissue samples to assist with the diagnosis of prostate cancer, such as with this section from a lymph node, where a PSA stain is highlighting a metastatic prostate adenocarcinoma in brown. Similar to this image at 40 times magnification, the prostate will have glandular structures called acini that are surrounded by supporting fibromuscular stroma. Each acinus has 2 layers of cells, an epithelial layer and a basal layer. The glandular epithelial cells can become hyperplastic and consist mostly of simple columnar and cuboidal cells, with some regions having pseudostratified columnar epithelium. The basal layer consists of smaller stem cells that rest on the basement membrane. The basal layer will not be seen in prostatic carcinoma. There are also neuroendocrine cells present, but they aren't easily identified with an H&E stain. With age, the acini have a higher probability of accumulating thick secretions that form hyaline spherical masses or concretions called corpora amylacea, which can be seen in these 3 alveoli. They can vary in size, but typically have layered or lamellated bodies that can be seen under the microscope. Over time, they may become partially calcified as well. The corpora amylacea are normal findings in the prostate and can be seen in normal lung tissue as well. The prostate gland is also divided into 4 main zones: the periurethral, transitional, central, and peripheral zones. Also, the anterior surface of the prostate consists of a dense fibromuscular stroma made of irregular connective tissue and smooth muscle that contracts during ejaculation. However, the prostate doesn't have a true distinct capsule itself. The central zone is the area that surrounds the ejaculatory ducts and comprises about 25% of the prostate's glandular tissue. The peripheral zone makes up 70% of the glandular tissue and surrounds the lateral and posterior sides of the central zone. This is the zone that's palpable during digital rectal exams, and is also the zone that's most susceptible to prostatic carcinomas as well as inflammation. The transitional zone comprises about 5% of the glandular tissue and contains the mucosal glands. This is the zone that undergoes hyperplasia in older individuals with benign prostatic hyperplasia, or BPH. When compared to a normal prostate's glandular epithelium, we can see that a prostate with BPH has significantly increased number of columnar epithelial cells that protrude further into the lumen, particularly in the upper right of this image. Hyperplasia of the supporting tissue or stroma can also be seen with BPH. Even though BPH leads to an increased amount of prostate tissue, it does not lead to prostatic carcinoma. The hyperplastic tissue compresses the prostatic urethra, causing difficulties with urination. And finally, the periurethral zone is a small zone surrounding the urethra, which contains mucosal and submucosal glands. As BPH progresses, this zone's stromal components may also increase in size and contribute to urethral compression. Although the different zones are not clearly separated histologically or anatomically, they are important clinically due to their associations with different pathological processes. Alright, as a quick recap. The prostate gland is a large, dense exocrine gland responsible for secreting a white alkaline seminal fluid that contains various molecules, fibrinolysin, citric acid, PAP, and PSA. The gland consists of branched tubuloalveolar glands and is divided into 4 zones: the periurethral, transitional, central, and peripheral zones. Prostate cancer is most closely associated with the peripheral zone, and BPH is typically associated with the transitional zone. Each zone has glandular epithelium that consists mostly of simple columnar or cuboidal cells, and a basal layer of stem cells that rest on the basement membrane. Older individuals have an increased number of corpora amylacea, which are hyaline spherical masses within some alveoli that may partially calcify over time. Now, let's get a closer look.
The pituitary gland is a small gland with a diameter of approximately 1 centimeter. It sits within a saddle-shaped bone cavity called the sella turcica, at the base of the brain. The gland is divided into 2 main parts: the anterior pituitary, or the adenohypophysis, and the posterior pituitary, or the neurohypophysis. Each part originates from different embryological sources, which is reflected in their different structure and function. Adeno means related to glands, and the adenohypophysis consists primarily of glandular epithelial tissue. In comparison, the neurohypophysis consists mainly of neural secretory tissue, since the neurohypophysis arises from a bud of nervous tissue that grows down from the hypothalamus. In this low-power parasagittal section of the pituitary gland, the anterior and posterior parts of the pituitary can be identified pretty easily when stained with Mason's trichrome. Generally, this stain will stain nuclei and basophilic structures blue. Collagen will be blue or green, and non-basophilic cytoplasm in red blood cells will be red. The posterior pituitary is composed of mostly unmyelinated axons, which do not have nuclei or a large amount of collagen. So as a result, the posterior pituitary doesn't stain well, and will look significantly lighter when compared to the anterior pituitary, whether it's stained with Mason's trichrome or hematoxylin and eosin. In between the anterior pituitary and posterior pituitary is a thin strip of cystic tissue, called the pars intermedia, which is part of the anterior pituitary because the cysts are actually remnants of Rathke's pouch, which is the embryological structure that eventually develops into the anterior pituitary. Zooming in closer to the pars intermedia, the colloid-filled cysts can be seen more clearly, along with irregular clusters of basophilic cells that often invade the neighboring neurohypophysis as well. Looking even closer, we can see a portion of the pars intermedia that has an area of lymphocyte infiltration. It's also easier to see that the adenohypophysis has a much richer blood supply compared to the neurohypophysis, with many small blood vessels present throughout the adenohypophysis, including the pars intermedia. Alright, let's move anteriorly and take a look at the adenohypophysis. Besides the bright red erythrocytes, there are 2 main groups of cells that are named based on how well they stain. The chromophils have cytoplasm that stains strongly, and as a result appear darker. The chromophobes have cytoplasm that stains weakly, and appear lighter in the image. The chromophils can then be divided into acidophils that stain dark red, and basophils that stain purplish blue due to their affinity for acidic or basic dyes, respectively. Acidophils consist of somatotrophs and lactotrophs. Therefore, they're responsible for secreting growth hormone, or somatotropin, as well as prolactin. The basophilic cells consist of corticotrophs, gonadotrophs, and thyrotrophs. so they secrete the remaining hormones associated with the anterior pituitary, including melanocyte-stimulating hormone, or MSH, adrenocorticotrophic hormone, or ACTH, luteinizing hormone, or LH, follicle-stimulating hormone, or FSH, and thyroid-stimulating hormone, or TSH. Let's move on to the posterior pituitary, which consists mostly of unmyelinated axons with neurosecretory activity. These axons can be seen throughout the image as the lightly stained portions of the tissue. The axons carry oxytocin and antidiuretic hormone, or ADH, from the hypothalamus down to their dilated nerve terminals called Herring bodies, which are neurosecretory bodies. The Herring bodies release either oxytocin or ADH, which is carried away by nearby capillaries. Also seen more clearly in this image are the highly branched pituocytes, which are the supporting glial cells. Alright, as a quick recap, the pituitary gland has two main parts, the anterior adenohypophysis and the posterior neurohypophysis. The anterior pituitary is made primarily of glandular tissue that's broadly split into chromophils and chromophobes. The chromophils are then further divided into basophils that stain purplish blue, and acidophils that stain dark red. The posterior pituitary is primarily neural tissue. Under high magnification, the distended nerve terminals called Herring bodies can be seen, which release either oxytocin or ADH to be carried away by nearby capillaries.
Now, let's get a closer look. The penis is composed of 3 cylindrical columns of erectile tissue, which are bound together by a dense fibroelastic layer called the tunica albuginea. In this very low-power transverse cross-section of the penis, the upper 2 columns are the dorsal corpora cavernosa of the penis, and the lower column is the ventral corpus spongiosum, which also contains the penile urethra. At the distal end of the penis, the corpus spongiosum expands to become the glans penis. The tunica albuginea is surrounded by a layer of superficial fascia that contains connective tissue, prominent blood vessels, and nerves. The dorsal side of the penis, which is the top side of this image, typically has the major blood vessels present, whereas the blood vessels within the columns of erectile tissue are significantly smaller. Surrounding the superficial fascia is the outermost sheath of penile skin, which can only be partially seen in the upper left of this image. The penile skin moves freely over the underlying tissues due to the loose hypodermis. Unless circumcised, it extends over the glans as the prepuce, or foreskin, which acts as a retractable protective fold of skin. If we take a closer look at the corpora cavernosa at 10 times magnification, we can see that the tissue is highly vascular, with a lot of red blood cells visible within the wide and irregularly shaped vascular sinuses. These sinuses are surrounded by walls or trabeculae that contain mostly elastic connective tissue and smooth muscle. If we zoom in a little closer, we can see a few cross-sections of smooth muscle that stain darker than the surrounding tissue. The sinuses are supplied by constricted, thick-walled arteries and arterioles called Helicine arteries. As their name suggests, the Helicine arteries are normally coiled when the penis is flaccid. But during an erection, following the parasympathetic stimulation, the smooth muscle surrounding the sinuses and arteries relax, causing the arteries to dilate and straighten out. This will lead to an increase in blood flow to the cavernous spaces, resulting in engorgement of the sinuses as well as compression of nearby veins, which helps further enlarge and maintain engorgement of the erectile tissue. Moving to the ventral corpus spongiosum, you can see that the erectile tissue looks very similar to the corpora cavernosa, except it contains the urethra in the middle. The corpus spongiosum also has a tunica albuginea that's more flexible or extensible. This allows the corpus spongiosum to expand a little more during an erection, which will help decrease compression of the penile urethra. Zooming in further, we can see more clearly that the urethra is also surrounded by a number of urethral glands called the mucus glands of Littré, and they are ducts that release a clear mucus directly into the urethra during an erection. If we take a closer look at the urethra, we can see the lining of the urethra better. The majority of the urethra is lined with pseudostratified columnar epithelium, which can be seen in this image. But the lining will be different in certain portions of the urethra as well. For example, the prostatic urethra has transitional epithelium. And distally, at the opening of the urethra, or urethral meatus, the lining changes to stratified squamous epithelium. Alright, as a quick recap, the penis contains 2 dorsal columns of corpora cavernosa and 1 ventral periurethral column of corpus spongiosum. All 3 columns contain erectile tissue that is comprised of highly vascular cavernous sinuses, smooth muscle, and helicine arteries. The urethra is primarily lined with pseudostratified columnar epithelium. Now, let's get a closer look.
The pancreas is a large gland that has both exocrine and endocrine functions. The majority of the pancreas consists of exocrine glands that produce about 1.5 liters of alkaline digestive enzymes daily, which is secreted directly into the duodenum. The pancreas also contains small endocrine cells found in clusters, called islets of Langerhans, which typically stain lighter than the pancreatic tissue around it. The pancreas has a thin collagenous capsule that surrounds the entire pancreas, although only portions of it can be seen in this image. The capsule also extends into the pancreas as septa, forming lobules. This section of the pancreas was prepared with Asin stain, which stains collagen a bluish-purple color in order to highlight structures like the capsule, septa, as well as the connective tissue that surrounds large blood vessels. Asin stain will also stain the connective tissue surrounding the interlobular ducts a similar color as well. But these ducts can be differentiated from the blood vessels by the presence of digestive enzymes instead of red blood cells within their lumen, as well as their distinct epithelium that lines the lumen. The epithelium consists of simple columnar cells in this image, but some ducts might also consist of stratified columnar or stratified cuboidal epithelium instead. To the right of the interlobular ducts is the adipocyte. The adipocytes actually increase in number within the pancreas as individuals age, which is a normal finding that's caused by pancreatic atrophy over time. The main functional tissue of the pancreas is called the parenchyma. If we take a closer look at this slide stained with hematoxylin and eosin, or H&E for short, we can see that the majority of the parenchyma consists of the exocrine portion of the pancreas. The exocrine secretory cells are arranged in groups that resemble a berry surrounding a central lumen. In Latin, acini means berries, which is why the groups of exocrine cells are called acini. We can see that the secretory cells are pyramid-shaped, and their nuclei are found slightly closer to their bases. This particular slide was stained with H&E, which allows us to see the purple, basophilic rough endoplasmic reticulum seen closer to the base of each secretory cell, and the pink eosinophilic secretory granules at the apices are inactivated enzymes called zymogens. In acute pancreatitis, it's the exocrine secretory cells that are damaged and end up releasing their enzymes into the immediate surroundings. These strong digestive enzymes cause additional damage to the pancreatic tissue that leads to severe inflammation as well. The center of the acini will sometimes contain one or more cells with a paler appearance. These cells are called centroacinar cells, and they're actually extensions of the small ducts that drain the digestive enzymes produced by the acini, called intercalated ducts. The presence of centroacinar cells is a unique feature of the exocrine pancreas, and is also an identifying marker that indicates the beginning of the intercalated ducts. These ducts are lined by simple cuboidal epithelium, and can be seen in this image as a linear arrangement of cuboidal cells. And in this cross-section of the intercalated duct, you can see the circular duct that's formed by the cuboidal cells. The intercalated ducts then drain into slightly larger intralobular ducts, like the one here. As the ducts progress, they gradually increase in diameter, and the connective tissue surrounding these ducts also thickens. The epithelium will also transition from simple cuboidal cells to stratified cuboidal cells. The intralobular ducts then drain into the interlobular ducts, which are located between the lobes but within the septa. These ducts are lined with epithelium that can be simple columnar, or stratified cuboidal epithelium, and have a very thick layer of connective tissue surrounding them. The interlobular ducts eventually join with the large main pancreatic duct, which runs the length of the pancreas and leads to the common bile duct in order to drain into the duodenum. A smaller accessory duct is also often present, which also drains into the duodenum. Let's move on to the endocrine portion of the pancreas, which forms clusters of paler cells such as the one seen in this image, called islets of Langerhans. The overall size of the islets can vary, but they can be identified pretty easily by their lighter stain and smaller cells when compared to the surrounding exocrine tissue. When we increase the magnification to 20x, we see that the fenestrated or perforated capillaries within the islets as well. The islets of Langerhans consist of 4 main types of cells, but they cannot be easily distinguished from one another on slides stained with H&E. Beta cells are the most common cell type, which comprises approximately 70% of pancreatic islets and secrete insulin. The islets also consist of alpha cells that secrete glucagon, delta cells that secrete somatostatin, and PP cells that secrete pancreatic polypeptide. This image of an islet was stained with Gamori stain, which allows us to differentiate the beta cells that stain bluish purple, and the alpha cells that stain pink. And we can also see just how much of the islet is made of beta cells. In individuals with type 1 diabetes, there'll be a decreased number of beta cells, as a result of autoimmune destruction of these cells specifically. Alright, as a quick recap. The pancreas has both exocrine and endocrine functions. Acini are the berry-shaped groups of exocrine cells that secrete digestive enzymes. The individual secretory cells are pyramid-shaped with basophilic rough endoplasmic reticulum and nuclei at their base, and eosinophilic granules at the apex. Islets of Langerhans are responsible for the pancreas's endocrine secretion, and are seen with H&E by their paler stain and smaller cells when compared to the surrounding tissue. The islets consist of alpha, beta, delta, and PP cells, with beta cells making up about 70% of the islets.
Now, let's get a closer look.
The nasal cavity is separated into 2 spaces, or fossae, by the nasal septum. The nasal septum can be seen along the midline. The nasal turbinates project into the nasal cavity in order to help increase the total available surface area. Both the paranasal sinuses, as well as the majority of the nasal cavity, are lined with respiratory mucosa. Only the roof of the nasal cavity is lined with olfactory mucosa instead. This image of a human's nasal mucosa at 10x magnification was prepared with Alchian blue and Van Giesen stains. If we zoom in to 40x magnification, we can see that the epithelium consists of pseudostratified ciliated columnar cells, along with many goblet cells that can be easily identified in light blue, because of the Alchian blue stain. This epithelium is also referred to as respiratory epithelium, and can generally be found in the conducting portions of the respiratory tract. The supporting connective tissue, called the lamina propria, is deep to the epithelium and contains many blood vessels and seromucous glands. If we compare the respiratory epithelium to the olfactory epithelium, we can see that the olfactory epithelium has the pseudostratified columnar cells, but it's significantly thicker and is composed of a combination of olfactory, sustentacular, and basal cells. The surface of the olfactory epithelium is also lined with modified cilia that function as olfactory receptors, which allows the olfactory cells to sense smells. Moving on to the larynx, this image is a low-power, coronal section of the larynx. There are 2 pairs of folds within the larynx. The upper folds are the vestibular folds, or false vocal cords, and the lower folds are the actual vocal folds, or the true vocal cords. Between the folds on each side are narrow spaces simply called ventricles. Each true vocal cord will need relatively large muscles in order to make the vocal cords for speech. These muscles, called the vocalis muscles, are stained darker within the vocal folds. The long structures on both sides of the larynx are portions of the thyroid cartilage, and the cricoid cartilage is partially seen at the bottom as well. Both of these support structures contain a large amount of hyaline cartilage that can be identified by their dark glassy appearance under higher magnification. The structure at the top of this image is the epiglottis. Now let's take a closer look at the upper vestibular fold, stained with hematoxylin and eosin, or H&E for short. Within the fold, there are many seromucous glands that help trap contaminants as well as increasing moisture in the air that's breathed in. The surface is lined with pseudostratified ciliated epithelium with goblet cells, or in other words, respiratory epithelium. When stained with H&E, the goblet cells can be identified as the clear oval structures within the epithelium. The vestibular folds might also have patches of stratified squamous epithelium in many non-smoking adults, but almost all smokers will have these patches. Moving down to the true vocal cords, we can zoom in and see that the portions of the vocal cords are lined with pseudostratified ciliated epithelium with goblet cells, or respiratory epithelium, similar to the upper vestibular folds. Between the epithelium and the vocalis muscle is a relatively thick ligament that runs the length of the vocal cords called the vocalis ligament. In this image, it's cut transversely and can be difficult to identify, but it's normally seen as a band of connective tissue just underneath the epithelium, near the apex of the vocal fold. The epithelium that covers this ligament is under a lot of mechanical stress when the vocal cords vibrate and hit one another in order to create sound during speech. So instead of respiratory epithelium, this portion of the vocal cords are lined with stratified squamous non-keratinized epithelium, which helps prevent physical damage to the surface of these folds. The remaining tissue between the epithelium and surrounding the vocalis ligament is the lamina propria, which consists of dense connective tissue with poor vasculature and no lymphatic vessels. Since the vocal cords are exposed to a lot of mechanical stress and harmful substances in the environment, there are quite a lot of conditions involving the epithelium of the vocal cords in particular. For example, vocal cord nodules, or singer's nodules, can form from frequent overuse or misuse of the voice. The nodules are benign reactive polyps that often form in the stratified squamous epithelium of the true vocal cords. Also, both cigarette smoke and alcohol consumption increase the risk of hyperplasia or dysplasia of the squamous epithelium within the larynx, leading to benign squamous papillomas, or invasive squamous cell carcinoma. And neonates can be infected by human papillomavirus, or HPV, from their infected mother during birth. This can lead to the development of laryngeal papillomatosis, where tumors grow in the infant's airway, which can cause respiratory difficulties and may eventually lead to suffocation if not treated. Alright, as a quick recap. The nasal cavity is mainly lined with respiratory epithelium, which consists of pseudostratified ciliated columnar cells with goblet cells. But the roof of the nasal cavity will have olfactory epithelium instead, which has a significantly thicker epithelium with modified cilia that function as olfactory receptors. The larynx consists of two folds, the vestibular folds and the vocal folds. The vestibular folds are primarily respiratory epithelium and have a large number of seromucous glands, while the vocal folds have respiratory epithelium as well, and stratified squamous epithelium that covers the vocalis ligament.
Now, let's get a closer look. Histology is the study of tissues and organs using microscopy. Similar to the phrase form follows function, the structures of human tissues are closely related to their functions. By integrating histology with other disciplines such as biochemistry, cell biology, and physiology, we can gain a much better understanding of how the body functions. While there are many types of microscopy, light microscopy, or LM, is the most common, largely because of how practical it is both in the clinical setting as well as in research. Other types of microscopy include scanning probe, ultraviolet, virtual, and electron microscopy, or EM. Light microscopy can be further subdivided based on the specific technique that's used, such as bright field, immunofluorescence, and dark field microscopy. We'll be focusing primarily on bright field microscopy and some of the various staining methods that go along with it. Bright field microscopy is not only the simplest type of light microscopy, but also the type that most people are familiar with, utilizing ordinary light to examine stained tissue at high magnification. In order to visualize the tissue, it needs to be properly prepared first. The most common method of tissue preparation has three main steps. First is to fix or preserve the tissue using formalin. The next step is tissue processing, which is the step that removes water from the tissue. This is done by using ethanol, which replaces the water in all the cells, and xylene, which removes ethanol by dissolving it, making it easier to embed the tissue in paraffin. Embedding the tissue in paraffin wax is the third step, which removes xylene from the tissue and also solidifies into a paraffin block that makes it easier to section, or cut the tissue into very thin slices. This is done using a machine called a microtome, which most labs use to cut the paraffin block into slices 4 micrometers in thickness. Next, the paraffin slice is mounted on a glass slide and the tissue is stained to make it easier to see under the microscope. The most common stain is the combination of hematoxylin and eosin, or H&E for short. This image of a pancreatic duct was stained with H&E, and the image on the right was stained with only hematoxylin. Hematoxylin is a positively charged basic dye, and will stain negatively charged structures purple or dark blue. Because of this characteristic, tissue components that strongly stain with basic dyes are often called basophilic structures. Nuclei, ribosomes, and rough endoplasmic reticulum are all basophilic structures because of the negatively charged DNA and RNA that they contain. The cartilage matrix is also a common basophilic structure as well. On the other hand, eosin is a negatively charged acidic dye that stains positively charged or eosinophilic structures red or pink. Some of the commonly eosinophilic structures include collagen, cytoplasmic proteins, and mitochondria. Mitochondria are especially eosinophilic and stain strongly to eosin, giving cells with a lot of mitochondria, like cardiac muscle cells, a dark pink coloration. As you might expect, other stains can be used in order to help visualize and examine specific structures that might not as easily be seen with H&E. The periodic acid Schiff, or PAS stain, is also commonly used. It stains complex carbohydrates dark red or magenta, making it useful for identifying glycogen in cells, such as hepatocytes and muscle cells. In this image of hepatocytes, the tissue was stained with PAS, and also counterstained with hematoxylin. This allows us to see the dark magenta glycogen, as well as the nuclei of the hepatocytes, which are stained purple with hematoxylin. The sugars found within mucus are also stained well by PAS, such as the mucus found lining the stomach, which is intensely stained a dark magenta in this image. The PAS stain will also stain connective tissue and basement membranes, such as the glomerular basement membrane and the basement membranes of Bowman's capsule found in the kidneys. This section was taken from the small intestine and stained with Masson trichrome. This stain is used to visualize supporting tissue better, particularly collagen. With Masson trichrome, connective tissue will stain blue, as we can see clearly in the submucosa of the small intestine. If we zoom in closer, we can see that nuclei stain dark red or purple, and cytoplasm will stain red or pink. Keratin, erythrocytes, and muscles will also stain red or pink as well. At this magnification, we can also see the lamina propria in blue, which is the connective tissue that supports the epithelium. Alcian blue stains acidic polysaccharides blue, such as the polysaccharides found in cartilage and certain types of mucin. It's often used along with other stains such as H&E or Van Giesen. The mucin contained within goblet cells can be easily seen in this image of the nasal mucosa, although the stain appears almost green or teal because Van Giesen was also used to stain the tissue, making it easier to see the tissue surrounding the goblet cells. Van Giesen is often used to stain blood vessels and the skin. This stain in particular can be useful to visualize an abnormal increase in collagen or to help differentiate smooth muscle from collagen within a tissue sample. It stains collagen pink, elastic fibers black, nuclei blue, and erythrocytes, muscle, cytoplasm, and fibrin yellow. Giemsa stain is a standard stain used for blood and bone marrow smears. Erythrocytes will appear pale pink, cytoplasm a pale blue, and nuclei a dark blue or violet color. Sudan black and osmium are dyes that will stain lipid-containing structures a brown or black color, such as the myelin around nerves and adipocytes. Also, this stain can be useful in diagnosing metabolic diseases that result in increased intracellular cholesterol, phospholipids, or glycolipids. Clinically, frozen sections are used for much faster results when needed. For example, during surgery, if the surgeon is removing a tumor, the surgeon may need to make sure all the unhealthy tissue is removed by having tissue samples analyzed quickly by a pathologist before completing the surgery. To do this, the margin of the surgically resected mass is sent to the pathology lab, where the mass is embedded in a compound called optimal cutting temperature compound, or OCT for short. It's then rapidly frozen in liquid nitrogen. This will quickly preserve and harden the tissue so that it can be sectioned in a machine called a cryostat, which is a type of microtome where the tissue can be maintained at a sub-freezing temperature. The slides are most commonly stained with H&E, methylene blue, and or PAS stains. This method is used primarily due to its fast turnaround time, which can be as fast as 10 minutes if performed by an experienced pathologist. Frozen sections are usually only used for urgent analysis because the quality of the slides produced from frozen sections are not as good as the typical formalin fixed and paraffin embedded tissue, which can also be called permanent sections. The permanent sections can take about 12 hours to 2.5 days to complete, depending on the tissue size and staining method used. These images of necrotic fibroadipose tissue with fungal organisms show an example of the potential difference in image quality between a frozen section and a corresponding permanent section, both stained with H&E. Alright, as a quick recap. The most common form of light microscopy is bright field microscopy, which most commonly uses H&E stains. Hematoxylin will stain nuclei, ribosomes, rough endoplasmic reticulum, and cartilage matrix a purple or dark blue color, while eosin will stain collagen and cytoplasm red or pink. Other stains can be used along with H&E in order to help visualize and examine specific structures that might not be as easily seen with H&E alone. If urgent analysis of a tissue is required, frozen sections can be used in order to provide results much faster. Now, let's get a closer look.
The mammary glands of each breast consists of about 15 to 25 sections called lobes, with each lobe containing many smaller structures called lobules. Each lobule consists of a large number of alveolar glands, which are the small sac-like structures that produce milk. The milk produced by the alveolar glands drain into the terminal ducts that join together to form the intralobular ducts. These ducts converge further and form the lactiferous ducts, which drain the lobes and eventually lead to the nipple. All of the lobules and ducts are also surrounded by fibrofatty tissue that provide a supportive and protective function. Before pregnancy, the mammary glands are considered to be in a state of inactivity, and they're structurally different from mammary glands that are in the active state. The active state occurs during pregnancy and during lactation after childbirth. During pregnancy, the alveolar glands and the duct system will grow in preparation to produce milk for a newborn baby. This low power image is an example of lactating mammary glands in the active state. A portion of a lobe can be seen surrounded by a thick layer of connective tissue that contains a small number of fat cells or adipocytes within the connective tissue as well. Within the lobe, thinner layers of connective tissue separate the lobe into lobules. In a neighboring lobe, we can also see a couple of large lactiferous ducts. These ducts are lined with a double layer of columnar or cuboidal cells with a surrounding layer of connective tissue. If we take a closer look at one of the lobules, we can see some of the smaller intralobular ducts that are typically lined with 1 to 2 layers of cuboidal cells that also have a thin layer of connective tissue that surround the ducts. In this image of a different lobule, we can see a good example of a few alveoli and a longitudinal cross-section of the terminal duct that's responsible for draining the milk produced by these alveoli. Both the terminal ducts as well as the alveoli are lined with either cuboidal or columnar secretory cells. The supportive tissue between the ducts and alveoli is called the intralobular stroma. It consists mostly of loose connective tissue, but it also contains small blood vessels, fibroblasts, plasma cells, a few adipocytes, and myoepithelial cells. Although the wavy connective tissue can be easily seen in this image, some of the other structures can't be identified as easily, since the intralobular stroma is typically very thin and sparse in mammary glands that are in the active state. Now, if we compare mammary glands in the active state to an image of an inactive or resting mammary gland, the most significant difference seen histologically is the nearly complete absence of the milk-producing alveoli, which we can see even in these low-power images. As a result, the majority of the resting mammary glands consist of connective tissue and adipose tissue. The lactiferous ducts will still be present, but they'll typically be smaller and will often appear flattened or collapsed. But the lining of these ducts are still the same, with a double layer of columnar or cuboidal cells. The general structure of the terminal ductules are also still the same in resting mammary glands, with a simple cuboidal epithelium and a surrounding intralobular space that consists of connective tissue, fibroblasts, myoepithelial cells, and in this image, we can also see a small blood vessel present in the upper right. Myoepithelial cells surround the terminal ducts, but they also surround the lactiferous ducts and the alveoli. During breastfeeding, increased oxytocin will stimulate these smooth muscles to contract and lead to milk ejection from the nipple. The myoepithelial cells are typically located between the secretory cells and the basement membrane, so they are also an important histological marker for the presence and progression of primary breast carcinomas. As a breast tumor progresses, the reduction or disappearance of myoepithelial cells may suggest a transition to an infiltrating or invasive status. Alright, as a quick recap, the mammary glands consist of alveolar glands that are organized into lobules, along with the terminal and intralobular ducts that drain the alveoli. The active state starts during pregnancy when the lobules increase in both size and total number, and the alveolar glands become the most prominent structures in the breast. The inactive or resting state is when an adult female is not pregnant or breastfeeding. In this state, the ducts are smaller and the secretory alveoli are undeveloped. In both states, the alveoli and terminal ducts are lined with cuboidal or columnar secretory cells. The intralobular ducts will have 1 to 2 layers of cuboidal cells, and the lactiferous ducts will have 2 layers of columnar or cuboidal cells.
Now, let's get a closer look. The lymphatic system is an essential part of the immune system, and it consists of a network of lymphatic vessels, tissues, and organs. The lymphatic vessels drain interstitial fluid, or lymph, from peripheral tissues back into the blood. Lymphoid tissue and organs contain a lot of lymphocytes and other white blood cells. The primary lymphoid organs include the thymus and bone marrow, and the secondary lymphoid organs include the tonsils, lymph nodes, spleen, and mucosa-associated lymphoid tissue, or MALT for short. The spleen is the largest lymphoid organ. It receives blood from the splenic artery and is the only lymphoid organ that primarily filters blood instead of lymph. It's an encapsulated organ that's typically about 12 centimeters in length, 7 centimeters wide, and 3 centimeters deep. The spleen's functional tissue, or parenchyma, consists of red pulp with small white nodules of lymphatic tissue scattered throughout, called the white pulp. Although, when looking at the spleen histologically after it's been stained with H&E, the red pulp is actually stained a combination of pink and purple, and the white pulp is stained dark purple because it contains a large number of basophilic nuclei. If we take a closer look at the outer edge of the spleen, the capsule that surrounds the spleen is seen as a dense layer of pink connective tissue. The connective tissue also forms short extensions into the spleen called trabeculae. The trabeculae will also occasionally surround arteries as they enter the parenchyma of the spleen. These trabecular arteries are branches of the splenic artery. The remainder of the tissue seen in this image is the highly vascular red pulp. Now, if we move to a different region of the spleen, we can see that there are areas that are more basophilic and stain mainly purple. These areas of lymphatic tissue make up the white pulp, which consists mostly of T cells or B cells. The trabecular arteries from the previous image branch even more and become central arterioles. Each central arteriole is surrounded by a periarteriolar lymphoid sheath, or PALS for short, which is a cylindrical mass or sheath of mostly mature T cells that surrounds the central arteriole. At certain points along the central arteriole, the PALS have localized expansion of tissue that form lymphatic nodules of primarily B cells called splenic follicles. The formation of the follicles actually displace the central arteriole, so as a result, central arterioles are not centrally located in splenic follicles. The lymphatic tissue surrounding the central arteriole is still primarily mature T cells, but the rest of the lymphatic nodule consists primarily of B cells. The eosinophilic center of the follicle is the germinal center, which contains activated B cells. Immediately surrounding the germinal center is the mantle zone, which is a dark and more dense circle of B cells and macrophages. And the outermost region that surrounds the mantle zone is called the marginal zone. This zone is not as densely packed and forms a thicker ring of B cells, macrophages, and dendritic cells. Alright, now let's focus on the red pulp, which appears noticeably more eosinophilic, or pink, because of the large amount of red blood cells present. About 70% of the red pulp by volume is composed of the parenchyma, and about 30% consists of the venous sinuses, or sinusoids. The venous sinuses are broad interconnected vascular spaces that are lined with specialized endothelial cells that filter the blood. Defective or abnormally shaped RBCs, such as spherocytes, won't be able to pass between the endothelial cells, and they'll eventually be removed by nearby macrophages. The endothelial cells have large round basophilic nuclei and have reticular or net-like fibers that surround the sinusoids to provide structural support, although they can't be seen with light microscopy alone. The venous sinuses will eventually join with one another and drain into the splenic vein. So, because of the size of the venous sinuses, the parenchyma of the red pulp actually looks like cords under the microscope, which is why they're also called cords of Billroth, although if the red pulp was seen in 3 dimensions, it would actually look more like a piece of Swiss cheese, with the holes representing the sinusoids and the cheese representing the parenchyma. The parenchyma is a combination of macrophages, lymphocytes, loose connective tissue, arterioles, and RBCs that move from arterioles to sinusoids. The arterioles in the red pulp are continuation, or branches of the central arterioles found in the white pulp. The arterioles are no longer surrounded by lymphocytes, and are also simply called pulp arterioles. Portions of the pulp arterioles are called sheathed arterioles, because they are surrounded by macrophages that help filter the blood. But there are also arterioles that don't have sheaths, and they're considered to be non-filtering arterioles. Now, let's get a closer look.
The gallbladder is a muscular, pear-shaped storage sac attached to the lower surface of the liver. In addition to being able to store about 50 milliliters of bile, it also concentrates the bile before secreting it into the duodenum after meals. Bile is produced by the liver and secreted into a network of intrahepatic bile ducts before reaching the common hepatic duct. The common hepatic duct then connects with the cystic duct, which allows the bile to flow into the gallbladder. When the gallbladder contracts, bile is pushed back out of the gallbladder through the cystic duct and flows into the common bile duct, which joins with the main pancreatic duct before emptying into the duodenum. Focusing on the gallbladder at low magnification, the gallbladder wall has 3 main layers: the inner mucosa, tunica muscularis, and its outer layer of connective tissue called the external adventitia or serosa. If we first take a closer look at the mucosa, we can see that the mucosa has many mucosal folds, or ruga, but they're not long enough to be considered villi. Some of the folds of the mucosa are deep enough to form the appearance of cross bridges when seen under a microscope, such as the 2 in this image. If we increase the magnification to 40 times, we can see that the mucosal epithelium is lined with simple columnar cells that are overlying the lamina propria, which contains dense, irregular connective tissue, many immune cells, and small capillaries. The gallbladder's mucosal folds may look similar to portions of the intestine, but can be distinguished by the gallbladder's lack of intestinal crypts at the base of the mucosal folds. In the bottom of this image, we can see a portion of the next layer underneath the mucosa, called the tunica muscularis. Here, we can see more of the tunica muscularis, or the muscular layer of the gallbladder. The muscles in this layer are grouped in bundles of smooth muscle, but they are actually randomly oriented. The contraction of these muscles results in the emptying of the gallbladder. At the top of this image is the mucosa, and underneath the tunica muscularis is the gallbladder's outer layer of connective tissue. The thick outer layer of connective tissue is different depending on the section of the gallbladder we're looking at. The portion of the gallbladder that's attached to the liver has an external adventitia that's meant to attach the gallbladder to the liver, which is why it's mainly composed of dense, irregular connective tissue. The portion of the gallbladder that's not attached to the liver is covered by an external layer called the external serosa. If we look at a section of the serosa, we can see that it's primarily made up of loose and irregular connective tissue with an outer membrane of simple squamous cells called mesothelium. In comparison to the adventitia, the external serosa will also have a higher number of other structures present within the connective tissue as well, such as large blood vessels, adipocytes, and lymphatic vessels. All right, as a quick recap, the gallbladder is a muscular, pear-shaped organ that stores, concentrates, and then releases bile into the duodenum. It has three main layers: the mucosa, tunica muscularis, and an outer layer of connective tissue. The mucosa is highly folded with cross bridges and a simple columnar epithelium. The muscular layer is made of bundles of smooth muscles that are randomly oriented, and the outer layer of connective tissue is referred to as either the adventitia or the serosa. The adventitia is found in the portions of the gallbladder attached to the liver and consists of dense connective tissue. The serosa is found in the portions not attached to the liver and consists of loose connective tissue and an outer mesothelium of simple squamous cells.
Now, let's get a closer look. The gastrointestinal, or GI tract, extends as a single tube from the esophagus all the way to the distal portion of the anal canal. Although different parts of the tract may appear to have very different structures and functions, the wall still maintains four main layers all throughout the GI tract: the mucosa, submucosa, muscularis propria, and either an outer serosa or adventitia. Even in this low-power cross-section of the esophagus, we can see the inner mucosa, submucosa, and muscularis propria, although the outer adventitia isn't present in this image. All four layers have variations of their structure and function in different regions of the GI tract, but the mucosa is the layer that typically has the most significant changes. The mucosa of the esophagus consists of three main layers. At 20 times magnification, we can see each of the layers more clearly: the epithelium, lamina propria, and muscularis mucosa. The thick epithelial layer lines the lumen of the esophagus and consists of stratified squamous non-keratinized cells, which has their typical appearance of flat, overlapping cells that are more flat as they move away from the base or basal cell layer. The lamina propria is a much thinner layer of dense, irregular connective tissue. It provides a supporting function to the epithelium, such as the blood vessels within the connective tissue that supply blood to the epithelium. The muscularis mucosa is the outermost layer of the mucosa and is comprised of smooth muscle. The muscle fibers in this image have a circular or dot-like appearance because the fibers run longitudinally, or in the same direction as the esophagus. The lower esophageal sphincter, or LES for short, is actually not an anatomical sphincter, which means histologically there is no well-defined thickening or muscle that controls the LES. Instead, it's considered a physiological or functional sphincter. But when this sphincter isn't functioning properly, it can lead to gastric acid reflux into the lower esophagus, which can cause heartburn. Also, over time, prolonged exposure to gastric acid can cause damage to the epithelium of the esophagus. The body attempts to adapt by transforming the normal stratified squamous cells of the lower esophageal lining into a mucus-secreting epithelium with simple columnar cells, which are better at protecting themselves from the gastric acid. This condition is called Barrett esophagus, which is a form of metaplasia. Unfortunately, though, this metaplastic change also increases the risk of developing an esophageal adenocarcinoma. These changes can also be identified by using an endoscope, where we'd see salmon-colored patches at the lower segment of the esophagus. The presence of both endoscopic and histologic changes are needed for the diagnosis of Barrett esophagus. The next main layer is the submucosa, which consists mostly of dense collagenous connective tissue that stains pink. But the submucosa also contains mucus glands, blood vessels, lymphatic vessels, and nerves. Within the connective tissue, there's also elastin fibers, which allows the esophagus to expand when food passes through it. The mucus glands are visible here, and they are small, compound, tubuloalveolar glands that secrete mucus that stains purple. There are typically more mucus glands present in the upper and lower thirds of the esophagus. The location of mucus glands within the submucosal layer is actually a unique feature specific to both the esophagus and the duodenum. The mucus is excreted through ducts that reach the mucosa's epithelium in order to help lubricate the surface. These ducts typically have cuboidal or stratified cuboidal cells. The next main layer is the muscularis propria, which varies depending on the portion of the esophagus we're looking at. But throughout the entire esophagus, there will be an inner circular layer of muscle and an outer longitudinal layer of muscle. The proximal portion of the esophagus is under voluntary control, so the upper third of the esophagus will have primarily skeletal muscle. The muscle type will gradually transition from skeletal to smooth muscle. So, as a result, the middle third will have both skeletal and smooth muscle present, and the lower third will consist entirely of smooth muscle. This cross-section of the esophagus was taken from the middle third, so if we zoom in to 40 times magnification, we can see that both skeletal and smooth muscle are present. The skeletal muscle can be differentiated from the smooth muscle by the typical striations we'd expect to see in skeletal muscle fibers. Between the inner and outer layers of muscle, there's also a network of nerves called the Auerbach's plexus, or myenteric plexus. In this image, a ganglion, or a group of neuron cell bodies, can be seen with its pink outer capsule visible as well. These nerves innervate the muscles, and are responsible for producing the organized rhythmic contractions that result in the peristaltic movement of the esophagus. The majority of the esophagus is surrounded by its outermost layer of connective tissue called the adventitia. In this image, the underlying muscularis propria can still be seen at the top. The adventitia is attached to nearby structures, such as retroperitoneal organs, which keep the esophagus in a relatively fixed position. The last 1 to 2 centimeters of the esophagus are within the abdominal cavity, after crossing the diaphragm. This portion of the esophagus has an outer layer of connective tissue called the serosa. The serosa is covered by a simple squamous epithelium, or mesothelium, that's continuous with the abdominal mesenteries. If we increase the magnification, we can see some of the additional structures within the connective tissue, such as a large artery, lymphatic vessels, and adipocytes. These structures would be found in both the adventitia as well as the serosa. Alright, as a quick recap. The wall of the esophagus has 4 main layers: the mucosa, submucosa, muscularis propria, and either the serosa or adventitia. The mucosa consists of 3 parts: the stratified squamous epithelium, its supporting lamina propria, and a thin layer of smooth muscle called the muscularis mucosa. The submucosa consists mostly of dense, irregular connective tissue, but also contains unique mucus glands that are only seen within the submucosa in the esophagus and the duodenum. The muscularis propria is the main layer of both smooth and skeletal muscle, with an inner circular layer of muscle and an outer longitudinal layer of muscle. The last 1 to 2 centimeters of the esophagus are surrounded by an outer layer of serosa, but the majority of the esophagus has an outermost layer of connective tissue called the adventitia. Now, let's get a closer look.
Cartilage is an avascular, smooth, and elastic tissue with various forms that allow it to function in a variety of ways. It can act as a smooth surface for bone articulation within joints, play a role in bone formation, and also provide structural support, such as the cartilage rings in the trachea. The majority of the tissue consists of extracellular matrix, or ECM, and only a relatively small number of chondrocyte cells. Mature chondrocytes are responsible for secreting and maintaining the extracellular matrix of the cartilage. Based on the appearance and specific function, cartilage is separated into three main types: hyaline cartilage, elastic cartilage, and fibrocartilage. Hyaline cartilage has a glassy appearance and can be found on many articular surfaces, as well as cartilage that provides structural support. Elastic cartilage contains a dense network of branching elastic fibers that allows it to be more flexible, which is why it's often found in structures that need to be firm yet still flexible. Some examples include the epiglottis, larynx, and the pinna of the external ear. And finally, fibrocartilage contains dense connective tissue with collagen fibers, as well as bands of extracellular matrix. This type of cartilage is found in structures such as the meniscus and intervertebral discs. First, let's focus on hyaline cartilage, which is the most common type of cartilage. This image is an example of hyaline cartilage on the articulating surface of a long bone, with the articulating surface located at the top of the image. The image on the right is a longitudinal section of the trachea, which is an example of hyaline cartilage providing structural support. Both images were stained with hematoxylin and eosin, or H&E for short. With this stain, the cartilage stains a dark purple due to its strong affinity for hematoxylin. If we take a closer look at the tracheohyaline cartilage, it has an outer layer of dense, irregular connective tissue surrounding the cartilage called the perichondrium. The perichondrium is also further separated into an outer fibrous layer and an inner chondrogenic layer. Within the chondrogenic layer, there are spindle-shaped cells that differentiate into small cells called chondroblasts as they migrate deeper into the cartilage matrix. These cells will start producing cartilage matrix and eventually mature into chondrocytes. The chondrocytes are found within small cavities called lacunae that are often arranged in clusters of 2 to 4 cells. The extracellular matrix, with its glassy appearance, is composed of mostly type II collagen and proteoglycan, which is an amorphous gelatinous substance, often referred to as ground substance. Although articular hyaline cartilage is very similar to tracheohyaline cartilage, a key difference between them is that articular cartilage does not have a perichondrium of connective tissue surrounding it. The lack of perichondrium ends up limiting the articular cartilage's ability to grow. Now, elastic cartilage is similar to hyaline cartilage, except the extracellular matrix contains its defining feature of a dense network of elastic fibers, which gives this type of cartilage its flexibility and elastic characteristics. This image from the pinna of the external ear is stained with Mason's trichrome and aldehyde fuchsin. The aldehyde fuchsin stain highlights the elastic fibers dark pink within the matrix of the cartilage. At 32 times magnification, we can see some general features of elastic cartilage that are similar to hyaline cartilage, such as the cartilage matrix, which stains blue, the chondrocytes within the lacunae, and also the perichondrium at the top, which is stained purple and dark blue. The last type of cartilage is fibrocartilage, which has a mixture of features from hyaline cartilage and dense connective tissue. And similar to articular hyaline cartilage, this type of cartilage doesn't have a perichondrium. The general structure of fibrocartilage can be seen even at low magnification, with alternating layers of purple hyaline cartilage matrix and thick layers of dense connective tissue that have type I collagen that stains pink. The fibers are arranged in the same direction as their functional stress to maximize their strength and resist deforming. Zooming into 40 times magnification, we can find chondrocytes within their lacunae in the basophilic matrix, similar to the other types of cartilage. Fibrocartilage also has scattered fibroblasts that are typically within the fibrous region of the tissue. These cells can be identified by their long and thin nuclei, such as the one seen in this image. Alright, as a quick recap, there are 3 main types of cartilage: hyaline cartilage, elastic cartilage, and fibrocartilage. The hyaline cartilage has an extracellular matrix with a glassy appearance and can be found in articulating surfaces of long bones and structures such as the trachea. And the articular hyaline cartilage doesn't have a perichondrium. Elastic cartilage is found in structures such as the pinna of the external ear and the epiglottis. The matrix of elastic cartilage is similar to hyaline cartilage, except it also has a dense network of elastic fibers, and it always has a perichondrium. And finally, fibrocartilage is found in structures like the intervertebral discs and meniscus. The tissue is a combination of hyaline cartilage and dense connective tissue. So, as a result, it contains more type 1 collagen and fibroblasts compared to the other types of cartilage. And, similar to hyaline articular cartilage, it doesn't have a perichondrium.
Now, let's get a closer look. There are 3 types of muscles: cardiac, skeletal, and smooth muscle. Each type has distinct functions as well as structural characteristics that can be identified histologically. Cardiac muscle makes up the majority of tissue found in the wall of the heart. Each mature cardiac muscle, or cardiomyocyte, is relatively short, with a length approximately 85 to 120 micrometers long, and a diameter approximately 15 to 30 micrometers. Histologically, cardiac muscles have quite a few unique characteristics that make it easier to differentiate them from skeletal muscles. Unlike skeletal muscles, cardiac muscle fibers are branched cells with only 1 to 2 centrally located nuclei. Also unique to cardiac muscles are the intercalated discs, which are the specialized junctions between neighboring cells that allows the cells to have synchronized contractions and pump blood out of the heart efficiently. Let's take a look at a longitudinal section of cardiac muscle cells that was stained with hematoxylin and eosin, or H&E for short. If we compare cardiac muscle cells to skeletal muscle cells, we can see that there are some key differences between the two muscle types. Skeletal muscle cells don't branch and have multiple nuclei that are peripherally located along the cell, whereas cardiac muscle cells are branched and usually have just 1 or occasionally 2 centrally located nuclei. If we look at a cross-section of the cardiac muscle, it's easier to see that the nuclei are centrally located. Both cardiac and skeletal muscles have cross striations that are perpendicular to the muscle fibers, but they're much more prominent in skeletal muscles. Also unique to cardiac muscle cells are, as we mentioned, the intercalated discs that connect the neighboring cardiomyocytes. With an H&E stain, the intercalated discs are seen as thin, slightly darker lines that are perpendicular to the direction of the muscle cell. In order to see the intercalated discs better, a section of tissue can be stained with a specialized stain such as phosphotungstic acid hematoxylin, or PTAH for short. Although both the cytoplasm and intercalated discs will stain purple, the intercalated discs will be a significantly darker purple in comparison to the surrounding cytoplasm. Whether the section of cardiac muscle was stained with H&E or PTAH, lipofuscin will be seen as the yellow-brown pigment granules found near some nuclei. These granules are residues of lysosomal digestion, and they're considered to be a sign of wear and tear that accumulate in cardiac muscle tissue with age. Now, let's get a closer look.
Bronchioles are air-conducting passageways that are typically less than 1 millimeter in diameter. The smallest bronchioles, called terminal bronchioles, are the most distal airways that are still a part of the conducting portion of the respiratory system. The terminal bronchioles then lead to the first portion of the airways that have a respiratory function, called respiratory bronchioles. These bronchioles then lead to the alveolar ducts, which are transitional airways that gradually become increasingly involved in gas exchange and lead to the alveolar sacs in their individual alveoli. If we compare images from a bronchus and bronchiole at low magnification, we can see that the larger bronchi has large supporting cartilage plates that are not seen in the smaller bronchiole. The bronchi also contain seromucous glands that are not present in bronchioles, but the bronchioles will still have a surrounding layer of smooth muscle present. The larger proximal bronchioles, similar to the one in this image, are called the primary bronchioles. If we zoom into 40x, we can see that the primary bronchioles have ciliated, pseudostratified columnar epithelium with goblet cells. When using a hematoxylin and eosin stain, the goblet cells will appear lighter than the surrounding epithelial cells, and as their name suggests, these cells are often shaped similarly to the top portion of a goblet. Moving distally through the airways, the bronchiole's diameter gradually decreases and the terminal bronchioles will have a diameter less than 0.5 millimeters. The epithelium also transitions from ciliated pseudostratified columnar epithelium to an epithelium that consists mostly of ciliated simple columnar and cuboidal cells, with exocrine club or clara cells instead of goblet cells. The clara cells can be identified by their tall columnar and non-ciliated appearance, as well as their dome-shaped apical ends that contain secretory granules. If a patient has bronchial asthma, there'll be histological changes to the bronchioles. Mucus can sometimes be found in the lumen of the bronchioles, which can lead to the formation of Kirschman's spirals, which are spiral-shaped mucus plugs that can be found in the sputum obtained from a tracheal and bronchoalveolar lavage. They originate from small bronchioles and are associated with chronic lesions that produce excessive mucus. Another change that can be seen as a result of bronchial asthma includes a thicker bronchiolar wall due to hypertrophy of the surrounding smooth muscle, inflammation, mucus gland hyperplasia, and enlarged blood vessels. Moving on to the first part of the respiratory portion of the respiratory system, we have the respiratory bronchioles. These bronchioles also have a mix of simple cuboidal and columnar epithelium with even more Clara cells. The bronchiolar walls are thinner compared to terminal bronchioles, but the main difference is that now they contain a small number of alveoli as well. Each respiratory bronchiole then branches further into alveolar ducts. Alveolar ducts are linear airways that lead to many alveoli that line the duct, with occasional regions that are lined with simple cuboidal epithelium. The alveolar ducts are also the most distal portion of the respiratory system to still contain smooth muscle around the epithelium. The alveoli have diameters about 200 micrometers wide and have small capillaries that allow red blood cells to be in close proximity with inspired air. In this image, there's also a large blood vessel on the right side. The alveolar surface epithelium creates the continuous lining for each alveolus and is made up of type 1 and type 2 pneumocytes. These cells also form the interalveolar septum that separates neighboring alveoli from each other. About 95% of the alveolar surface area is composed of type 1 pneumocytes, which also forms the alveolar part of the air-blood barrier. These cells can be identified by their large, flat appearances and elongated nuclei. Their cytoplasm is very thin, which can be less than 80 nanometers in thickness, and the cells form tight junctions between themselves and neighboring cells. The type 2 pneumocytes are cuboidal cells and only make up about 5% of the alveolar surface area. These cells are usually located near the intersections of the septums and also form tight junctions with neighboring cells, similar to type 1 pneumocytes. Type 2 pneumocytes are capable of synthesizing pulmonary surfactant and dividing in order to regenerate both types of pneumocytes. As a result, hyperplasia of type 2 pneumocytes is an important marker for alveolar injury and repair within the lungs. Alveolar macrophages are present in both the connective tissue of the interalveolar septum as well as the lumen of the alveoli, as seen in this image. The alveolar macrophages act as the main phagocytes of the alveolar surface. As a result, smokers, coal miners, stonemasons, and people living in urban areas will often have alveolar macrophages that are filled with carbon particles called anthracotic pigments and birefringent silica particles when slides are examined under polarized light. In both of these example images, the individuals had a long history of cigarette smoking. Alright, as a quick recap. The larger primary bronchioles have an epithelium made of ciliated pseudostratified columnar cells with a small number of goblet cells. The goblet cells can be identified by their lighter appearance and their goblet-shaped cells. Distally, the airway's diameter decreases and the epithelium transitions to ciliated simple columnar and cuboidal cells with Clara cells. The Clara cells can be identified by their tall, non-ciliated columnar appearance and their dome-shaped apical end. The respiratory bronchioles have a similar composition as the terminal bronchioles, except that they have a smaller number of single alveoli in their wall. The alveolar surface epithelium is made of type 1 and type 2 pneumocytes. Type 1 pneumocytes are large flat cells with elongated nuclei that compose about 95% of the surface area. Type 2 pneumocytes are cuboidal cells located near the intersections of the alveolar septums and will be hyperplastic when there's alveolar injury.
Now, let's get a closer look.
Bones are composed primarily of an extracellular calcified material called the bone matrix, or collagen matrix. It has three main types of cells: osteocytes, osteoblasts, and osteoclasts. Osteocytes are found in cavities, or lacunae, between the layers of the bone matrix and assist with the nutrition of the bone. Osteoblasts are found mostly along the surface of bones, but also within the bone matrix itself. They are responsible for both synthesizing and mediating the mineralization of the bone matrix. Osteoclasts are large multinucleated cells that are responsible for removing calcified bone matrix and allowing for the constant turnover and remodeling of bones. Morphologically, there are two main types of bones. The compact, or cortical bone, is the dense portion that's found closer to the surface of bones. The deeper portion of bones have interconnecting cavities and are called the cancellous, or trabecular bone. In this electron microscopy, or EM, image of the tibia, the compact bone is also blue, and the more central trabecular bone is yellow. Microscopically, both compact and trabecular bones will be organized or arranged in two forms. Layered or lamellar bone has a bone matrix that's arranged in sheets. 80% of lamellar bone is found within the compact bone, and only 20% is found within the trabecular bone. The more immature woven bone has collagen fibers that are arranged randomly. This form of bone is mostly found within developing and growing bones, as well as bones that have healed after being fractured. Alright, let's compare sections of a long bone and a flat bone. This long bone is a partial cross-section of the head of a long bone, and the flat bone is a cross-section from the skull. Both sections have been decalcified in order to make it easier to cut the bone into thin sections, but the collagen is still present in the slides. Since the denser compact bone is typically found closer to the surface, it's seen in the long bone along the outer edge, whereas the flat bone has compact bone on both of its outer flat surfaces. Trabecula means little beam in Latin, which is why the trabecular bone has small beam-like structures that are also called spicules. The spicules connect with each other to form a network of interconnecting spaces that contain bone marrow. The vertebrae is another example of a bone that has both types of morphology, but its compact bone is only a very thin outer layer, with the majority of the bone consisting of trabecular bone. In this image, we can also see that the bone marrow is still present within the spaces formed by the spicules. The osteoclast can be found within the bone marrow. In this image, the osteoclast is sitting within a resorption cavity it formed on the surface of the bone matrix. Identifying the multiple nuclei within the osteoclast can help differentiate it from other similar-looking cells like megakaryocytes, which only have a single multi-lobe nucleus. Okay, let's take a closer look at compact bones, which have structural units called the Haversian system, or osteons. An osteon consists of concentric lamellae, or layers, with a diameter of about 100 to 250 micrometers and a central canal called the Haversian canal. This canal contains small blood vessels and nerves, as we can see in this image. We can see a small blood vessel and remnants of nerve fibers as well. The concentric layers of mineralized bone matrix radiate out from the canal and are called the Haversian lamellae. Along the border of each osteon is a thin purple or basophilic line that's called the cement line. The white spaces throughout the osteon are the lacunae, which contain remnants of osteocytes. In this image of a Haversian canal, the osteoblasts can be seen lining the inner surface of the Haversian canal. Osteoblasts are large spindle-shaped or cuboidal cells that have a basophilic cytoplasm. Alright, as a quick recap. Morphologically, bones have two main types. The denser, compact, or cortical bone, which is found closer to the surface, and the cancellous, or trabecular bone, which is the spongy bone that consists of trabecula, or spicules, that form interconnecting cavities. Microscopically, the lamellar bone is arranged in sheets of collagen matrix, and woven bone has collagen fibers that are arranged randomly. There are also three main cell types that are part of bone. Osteocytes are found in cavities or lacunae between the layers of collagen matrix. Osteoblasts are found along the surface of bones and also lining the Haversian canal within the bone. And finally, osteoclasts are large multinucleated cells that can be found on the surface of the bone matrix.
Joints are structures in which bones meet to either permit movement or provide stability. There are plenty of them in our upper limbs. We've got the sternoclavicular, the acromioclavicular and the glenohumeral joints in the pectoral girdle, the elbow, the proximal and distal radio-ulnar joints in the forearm, the wrist joint, and the joints of the hand, which include the carpometacarpal, the intermetacarpal, the metacarpophalangeal, and the interphalangeal joints. So let's get acquainted with the first two of these joints. First up, there's the sternoclavicular joint, which is the only bony articulation attaching the upper limb to the axial skeleton. It involves the clavicular notch of the manubrium, the medial end of the clavicle, and a small part of the first costal cartilage. It is a saddle synovial type of joint, named so because the manubrial articulating surface is concave and the clavicular articulating surface is convex, so they fit together the same way that a cowboy sits on his horse's saddle. The joint has an articular disc that is firmly attached to the anterior and posterior fibrous joint capsule by the anterior and posterior sternoclavicular ligaments, as well as the interclavicular ligament. Like any other synovial joint, the articular surfaces of the sternoclavicular joint are covered by the fibrous joint's capsule, which has 4 ligamentous thickenings, simply called ligaments, lined internally by a thin layer of synovial membrane. The ligaments of the fibrous capsule are the anterior and the posterior sternoclavicular ligaments, which reinforce the joint capsule anteriorly and posteriorly, the interclavicular ligament, which strengthens the sternoclavicular joint superiorly, and finally there is the costoclavicular ligament, which anchors the inferior surface of the medial end of the clavicle to the first rib and its costal cartilage, providing stability to the sternoclavicular joint by limiting the elevation of the pectoral girdle. And second, there's the acromioclavicular joint. It lies between the lateral end of the clavicle and the tip of the acromion of the scapula. It's a plain type of synovial joint, meaning its articular surfaces are almost flat. Usually, there is a wedge-shaped articular disc between the articulating surfaces. The acromioclavicular joint is surrounded by a tough fibrous joint capsule with an internal synovial membrane that lines it. The acromioclavicular ligament strengthens the acromioclavicular joint superiorly and extends from the acromion to the clavicle. The acromioclavicular ligament alone can't hold this joint though, so it's reinforced by other ligaments further away from the joint, which are known as extrinsic ligaments. First, there is the coracoclavicular ligament, which is in fact 2 ligaments combined together to strongly anchor and fix the clavicle to the coracoid process. These are the conoid and the trapezoid ligaments. The conoid ligament is shaped, well, like a cone. Its base is attached to the conoid tubercle located at the inferior surface of the clavicle, and the apex attached inferiorly to the root of the coracoid process. And the trapezoid ligament, you guessed it, looks like a trapezoid. So no surprises here. It's attached to the superior surface of the coracoid process, from where it extends laterally to the trapezoid line on the inferior surface of the clavicle. Alright, before we wrap up, feel free to hit the pause button and see if you can remember the ligaments of the sternoclavicular and acromioclavicular joint. Alright, as a quick recap, the sternoclavicular joint is a saddle synovial type of joint, and it's the only bony articulation that attaches the upper limb to the axial skeleton. It has 4 ligaments, the anterior and the posterior sternoclavicular ligaments, which reinforce the joint capsule anteriorly and posteriorly, the interclavicular ligament, which strengthens the sternoclavicular joint superiorly, and the costoclavicular ligament, which provides stability to the sternoclavicular joint by limiting the elevation of the pectoral girdle. The acromioclavicular joint is a plain type joint that lies between the lateral end of the clavicle and the tip of the acromion. It is supported by the acromioclavicular ligament and the coracoclavicular ligament, which is made up of the conoid and trapezoid ligaments. The radio-ulnar joints are 2 joints between the 2 bones of the forearm, the ulna on the medial side and the radius on the lateral side. There's one superior or proximal radio-ulnar joint and one inferior or distal radio-ulnar joint, and together they allow for the movements of pronation and supination. This is going to be short and sweet, so let's dive right in. The proximal radio-ulnar joint is a pivot type of synovial joint, which consists of the head of the radius articulating with the radial notch of the ulna. The radial head is tightly bound into the radial notch of the ulna by the annular ligament that secures it in place. The two articulating surfaces are covered by a synovial membrane, over which lies the joint capsule. The joint capsule of the proximal radio-ulnar joint is an extension of the elbow's joint capsule. Next up, in the distal forearm, the radius and the ulna form another pivot type of synovial joint called the distal radio-ulnar joint, which is also covered by a fibrous joint capsule lined internally with a synovial membrane. For this joint, the rounded head of the ulna articulates with the ulnar notch of the distal radius. The articulating surfaces are mainly bound together by a fibrocartilaginous structure called the articular disc, which is triangular in shape, so it's also known as the triangular ligament. The articular disc attaches to the edge of the ulnar notch of the radius and the base of the styloid process of the ulna. The distal radio-ulnar joint is reinforced by the two important ligaments, the anterior and the posterior ligaments. These ligaments extend transversely from the radius to the ulna across the anterior and posterior surfaces of the joint. Both the proximal and distal radio-ulnar joints are responsible for forearm pronation, or the turning of the palm downwards on a flexed elbow, and supination, which is turning the palm upwards. To remember this, think about cupping both your hands and bringing them together to hold a bowl of soup, which mimics the movement of supination. For both these movements, the ulna remains relatively fixed and the radius moves over it. During pronation, the radial head rotates medially within the annular ligament at the proximal radio-ulnar joint, while the distal end of the radius is crossing over the ulna anteriorly and medially at the distal radio-ulnar joint. Pronation is done primarily by the pronator quadratus muscle, with a little help from the pronator teres. During supination, the radial head rotates laterally at the proximal radio-ulnar joint and causes the radius to uncross the ulna at the distal radio-ulnar joint, making the bones parallel again. Supination is done by two muscles, the supinator and the biceps brachii. Alright, as a quick recap, the proximal radio-ulnar joint is a pivot type of synovial joint, which consists of the head of the radius articulating with the radial notch of the ulna. The distal radio-ulnar joint is also a pivot type of synovial joint, and it consists of the rounded head of the ulna that articulates with the ulnar notch of the distal radius. Both the proximal and distal radio-ulnar joints are responsible for forearm pronation, or the turning of the palm downwards on a flexed elbow, and supination, which is turning the palm upwards.